You know, we usually think of our DNA as this super stable, unchanging blueprint, right? A fixed text that gets passed down from one generation to the next. Well, today we're going to flip that idea on its head. Turns out, huge chunks of our genetic code aren't static at all. They're actually mobile, dynamic, and can literally jump from one place to another inside our chromosomes. Okay, let's dive into the absolutely fascinating world of these jumping genes. So, what do speckled corn and nearly half of your own DNA have in common? I know, it sounds like the beginning of a weird biology joke, but the answer connects a really strange observation in a cornfield way back in the 1950s to the very core of what makes us who we are today. And that common thread? It's a process called transposition, driven by these things we call jumping genes. So, our story really kicks off with the brilliant scientist Barbara McClintock. While she was meticulously studying the chromosomes of corn plants, she kept noticing something that, well, it just didn't add up. Certain strains of corn had these kernels with a really unusual speckled pattern, mostly colorless, but with these vibrant patches of red. This wasn't some simple, predictable inheritance pattern. Something else, something dynamic and kind of unpredictable, had to be going on. Now, you gotta remember the scientific mindset back then. The genome was seen as this fixed, stable, almost sacred text. So when McClintock proposed that pieces of DNA could actually move, it was radical. I mean, it was almost unbelievable to a lot of her peers. She was basically suggesting the genetic blueprint could edit itself. It actually took decades for the rest of the scientific community to really catch up, but she was eventually awarded a Nobel Prize for this absolutely groundbreaking work. So how did she explain those speckles? Her explanation was just so elegant. It's basically a three-step process. First, this mobile element, she named Ds for dissociation, it jumps into the gene that makes the red pigment. That basically breaks the gene, turning it off. That's why you get the colorless background. But then, and this is the cool part, as the kernel is growing, in a few random cells, that Ds element would jump out again. When that happens, the gene is repaired, and it gets reactivated. Those cells, and all the cells they divide into, can now make the red pigment, creating those little speckles. It's a perfect visual of the jumping in action. McClintock's work really blew the doors open. Today, we know there are two main ways these transposable elements, or TEs for short, actually move around the genome. So let's get into the nitty-gritty of how they pull it off. The easiest way to think about these two mechanisms is with a simple analogy. Over on the left, you've got simple transposition. This is basically a cut and paste job. The element gets literally cut out of its original spot and pasted into a new one. Then on the right, you have retrotransposition, which is more of a copy and paste. Here, the original element stays right where it is, but the cell makes a copy of it, and that copy gets inserted somewhere else. And that's a crucial difference, right? Because copy and paste means the number of these elements can go up and up and up. Okay, let's zoom in on the cut and paste method. The star of the show here is an enzyme called transposase. First thing it does is it recognizes and grabs onto specific DNA sequences at both ends of the transposon. Then, it acts like a pair of molecular scissors, snipping the element right out of the chromosome. Transposase then carries this little piece of DNA to a new spot, makes a staggered cut in the target DNA, and slots the transposon in. The cell's own repair crew comes in to clean up the gaps, and in doing so, it creates these little identical sequences called direct repeats on either side of the new element. Pretty clever. Now for copy and paste. This pathway is a little more involved, and it actually uses a trick you might recognize from the world of viruses. First, the retrotransposon's DNA is transcribed into an RNA copy. Then, this amazing enzyme called reverse transcriptase does exactly what its name says. It reads the RNA and works backwards to build a brand new DNA version. Finally, another enzyme, integrase, takes this new DNA copy and, you guessed it, integrates it into a new location in the genome. And because the original never left its spot, this process can cause the number of these things to just explode. So we've got these moving elements, and we've got the different ways they move. But what do they actually look like on a DNA level? Well, geneticists can actually spot different types of these TEs based on their unique structures and the molecular tools they have packed inside. This table breaks it down nicely. First, you have the cut and paste simple transposons. Their telltale sign is these inverted repeats at each end, which are the landing pads for the transposase enzyme. Then you have the copy and paste retrotransposons. One group, the LTR retrotransposons, kind of look like retroviruses. They have long terminal repeats at their ends and use reverse transcriptase and integrase. The other group, non-LTR retrotransposons, don't have those big repeats, but they still use reverse transcriptase, along with another enzyme called an endonuclease to help them slice into the DNA. And this brings us right back to McClintock's corn. See, she actually found two related elements, which she named AC for activator and DS for dissociation. This slide explains the key difference. AC is what we call an autonomous element. It's got the full toolkit. It contains the gene for the transposase enzyme, so it can move all by itself. DS, on the other hand, is non-autonomous. It has the right signals at its ends to be moved, but its gene for transposase is broken. It's basically a stranded car that can only move if an autonomous AC element somewhere else in the genome comes along and provides the driver, that is, the enzyme. Okay, so we've seen how they were discovered and the mechanics of how they move. But so what? What's the bigger picture here? Why does it matter that these little bits of DNA are hopping around? Well, as it turns out, it matters a whole lot. Let's just start with this one. Staggering number, 45%. Almost half of your DNA is made up of these transposable elements and their ancient leftovers. No, that's not a typo. To put that in perspective, all the genes that actually code for proteins, that's only about 2% of our genome. This number alone tells you that these jumping genes are a massive feature of our genetic landscape. And we are definitely not alone. As you can see here, the corn genome is a whopping 60% transposable elements, which helps explain why McClintock was able to find them there in the first place. But the amount varies like crazy between species. Look at yeast, only 4%. This huge variation just begs the question, what is all this mobile DNA actually doing? One of the big ideas out there is the selfish DNA hypothesis. This theory frames these transposable elements not as something useful for us, but more like successful genetic parasites. The idea is that they don't exist to help us out. They exist simply because they are very, very good at making copies of themselves. This view kind of challenges that neat idea of a genome being a perfectly optimized machine. It suggests it's more like a dynamic ecosystem, where some of the residents are really just looking out for number one. But of course, it's not quite that simple. This whole process is a powerful force with both good and bad consequences. It's a classic double-edged sword. On the one hand, a TE jumping into a vital gene can be disastrous, causing diseases like hemophilia or even cancer. They can shuffle our chromosomes all around. But on the other hand, they're a major source of innovation. They can spread useful traits like antibiotic resistance in bacteria, they can tweak how our genes are regulated, and they can even create entirely new genes by grabbing bits and pieces of other genes and stitching them together. So we're left with this really profound question. For decades, a lot of this mobile DNA was just written off as junk DNA. But now we know it's an active, powerful, and massive part of our genomes. So what are they? Are they just genetic junk, leftovers from our evolutionary past? Are they ancient parasites that we've just been carrying along for the ride? Or are they the secret, chaotic architects of our own evolution? The real answer is probably a bit of all three, and scientists are still working every day to unravel the complete story.